Hello, dear students. In the last lecture of cytoplasmic inheritance, we discussed the example of maternal inheritance in Lemnia peregrina or gastropod shell coiling. Today, we will discuss the extranuclear inheritance of plastids in Mirabilis jalapa or 4 o'clock plant. As we all know that plastids are the membrane-bound cell organelles present in cytoplasm of plant cells. These are the autonomous bodies as they have their own circular DNA. These DNA have genes that can code for proteins. Now the plastids are of various types: chloroplast, leucoplast and chromoplast. The chloroplast contain chlorophyll and they are concerned with photosynthesis. Leucoplasts are colorless and they are concerned with storage. Similarly, chromoplasts contain colored pigments and they are associated with storage. Since egg contributes a large amount of cytoplasm in an individual, hence the plastids are inherited in majority by a female parent in many cases. This causes variegated conditions in various plants. Example, many colors in leaf branches, green, pale, red, pink colored patches in plants in a same plant. In some plants, this variegation is due to the nuclear genes, but in some plants like Mirabilis jalapa 4 o'clock plant, it is totally dependent on the cytoplasm or the egg cell, and this is called the maternal inheritance or cytoplasmic inheritance. Carl Correns 1909 observed this phenomena in Mirabilis jalapa. Carl Corren noticed variegate in Mirabilis jalapa. The variegated plant has three branches: a green branch, a pale white branch, and a variegated branch. The green branches have chloroplasts, the pale white branches contain leucoplast, and the variegated branches contain a mixture of chloroplast and leucoplast, which gives a mosaic appearance to that branch. Now the flower on a green branch produces an egg with chloroplast in its cytoplasm. The female flower on a white branch or pale branch contains an egg or develops egg having leucoplasts. The variegated branch forms the flower, and these flowers have the eggs of three kinds. Some eggs have only chloroplasts, some have leucoplasts, and some eggs have a mixture of chloroplast as well as leucoplast. On fertilization, he observed that a flower bearing on a green branch having an egg with chloroplast forms a zygote which produces a green colored plant irrespective of the type of pollen from any branch, green, pale or variegated. Similarly, a flower from a pale branch when fertilized formed the zygote having leucoplasts in it, but during the development such plant perished due to the lack of chlorophyll. The eggs of variegated branch, which were of three kinds, when fertilized with any type of pollens, formed three types of zygotes. The green chloroplast containing egg formed a green individual. The leucoplast containing egg fertilized into a pale plant which perished, and the variegated egg, the egg having a mixture of chloroplast as well as leucoplast, developed into a zygote containing both green chloroplast and pale leucoplasts. Now, in case of the zygotes containing both leucoplast and chloroplast, he observed another phenomena of cytoplasmic segregation, which is as follows. Here is a diagram to show the cytoplasmic segregation. This is a zygote which is obtained by the fertilization of a variegated flower having an egg containing both chloroplast and leucoplast, which upon mitosis develops similar cells as like the mother cells, which on further mitosis during the phase of development forms another cells. In some cases, cytoplasmic segregation happens to occur where leucoplasts segregate separately and chloroplasts segregate separately in two different cells. Segregation of leucoplast and chloroplast in pure cell lineages form two types of branches. These cells produce pale branches, and these cells which contain chloroplast forms green branches. And the branches and the cells which have both chloroplast and leucoplast, they produce variegated branches in the same plant. According to him, since the pollen contributes a little or no cytoplasm, hence the phenotype of the progeny is determined by the phenotype of the female parent. In a nutshell, if we represent eggs by a central circle, by the type of pollen outer circle, and zygotes in external circle, there is a three-circle diagram. One portion is representing those eggs which contain chloroplast or which are derived from green branches, other from pale branches, and the third one from variegated branches. If we fertilize these eggs with a pollen from green branch or pale branch or variegated branch, we will always obtain the zygotes having chloroplasts and forming green branches or green plants. If an egg of pale branch gets fertilized with a green or a pale or a variegated branch pollen, this will always produce the zygotes containing leucoplast only, and such plant will not develop further due to the lack of chloroplast. If an egg of variegated branch is fertilized with a pollen of green branch, pale branch or variegated branch, it will form hence three types of zygotes, one with chloroplast forming green branch, one with leucoplast forming pale branch, and one with variegated chloroplast plus leucoplast containing branches forming variegated plants. Similarly, here three types of plants and three types of plants will be formed. Correns also observed a surprising feature that if we do a reciprocal cross, the result will be different from Mendelian result. Means if we take a female green branch and a male white branch, then fertilization will give a green zygote, and it's reciprocal where we take a female as a white from white branch and male from green branch, it will form a pale white zygote. This is quite different from the result which we get from Mendelian crosses. Hence, Corren observed that the phenotype of a female actually determines the phenotype of a progeny. This is called a uniparental transmission, since the zygote is getting the phenotype from her mother, from its mother, hence this is called a maternal inheritance. And this is due to the autonomous bodies, plastids, which are inherited through cytoplasm of the plant cells. In the next lecture, we will discuss the kappa particles in paramecium in this chapter.